So here we will study about the most important concept of the molecular biology. That is, what is TCID₅₀? How do scientists measure viruses that does not form plaques? The answer is TCID₅₀. TCID₅₀ stands for tissue culture infection 50%. It is the amount of viruses needed to infect 50% of the culture cell or wells. Scientists prepare serial dilutions of the virus and infect multiple wells. After a few days, they observed cytopathic effect, that is CPE, changes or deaths of the cells caused by the viruses. Using methods like Muench or Spearman Carver, they calculate the virus concentration that infects 50% of the wells. Why it is important? Because it measures infectious viruses when plaques cannot be counted. So it is widely used for influenza, Japanese encephalitis virus, dengue, and many other viruses. So it helps standardize viral stocks for experiments. That's why it is important. Remember, PFU counts plaques while TCID₅₀ estimates infections in 50% of the cell cultures. Follow us for more molecular biology concept and please like and subscribe the channel. If you like the video, please save the video and watch later because you may miss the important concept. Okay, thank you.